It's my honor and pleasure to introduce tonight's speaker, Dr. Bruno Passarelli. Many of us have been waiting for this talk for a long time. Bruno is a professor at the Department of Biological Sciences at Saddleback College in Mission Viejo. Born and raised in Brazil, he moved to California to study music. After taking a marine biology class from a beloved professor named Ed Tarvid, he changed his major to biology and never looked back. He then earned an AA degree in general science from Santa Monica College, a bachelor's in marine biology, and a master's in biology from Cal State University, Long Beach, and a PhD in biology from U.C.L.A. Dr. Passarelli has been teaching a variety of courses at Saddleback College since 2011, such as marine biology, coastal ecology, evolution, and oceanography. He has also been leading study abroad programs to Brazil and Costa Rica since 2015. His research interests include marine invasive species and marine parasites, especially those associated with marine fishes. Tenho muito orgulho de você, meu amor. A mosca bebelete. The title of tonight's talk is Do Parasites Know Where the Grunion Go? Thank you, and please help me welcome Dr. Bruno Passarelli.
Can everybody hear me? All right, great. Julie, thank you so much for the introduction. It is my honor to be here at Cabrillo. This is a very special place for me. I want to tell you a little bit about my first visit to Cabrillo Marine Aquarium. Was almost 30 years ago. And as Julie mentioned, I was taking marine biology at Santa Monica College with Ed Tarvid. And another special professor who is here tonight, John Moss, is here. And they told me, they suggested that I come to Cabrillo to check it out. So I came here for an assignment, and I was fascinated. Like, I think a lot of you guys seen the same, going through the exhibit hall. I remember it looked like a living textbook. That's how I would describe it. Back in the day, 30 years ago, I didn't have an iPhone, not even a flip phone, but I had a notebook, so I was taking notes. And I remember I'm hearing that the aquarium was going to close, and I was like, man, I need to take notes. And I kept trying to write as much as I could. And then I finally calmed down and said, okay, I need to come back. Like, this place is so cool. There's so much to see. And I've came back so many times. So this is a very special place, and I'm happy to share some of the things that I know. Now, a few of you know that I'm Julie's husband, and some of them said, oh, so Julie got you, right, to give the talk. No, she asked me, right? We talked about that. It kind of went like this. A few months ago, like she said, she prepares way ahead of time. One day I came home, and she said, hey, you were giving the talk on April 3rd. It's already booked. Okay. So I guess she asked me, right? And I said, sure, it's okay. But I'll tell you one thing. Julie has been hosting the lectures here, and but one thing that I'm sure a lot of you know, Julie also do amazing research here at Cabrillo Marine Aquarium, and she had a nice question to me. I have a nice question for you guys. How would you like to see Dr. Passarelli, Julie Passarelli, give a talk about her research here at Cabrillo? All right, cool. Reactions from home. That's awesome. That's awesome. All right, so we'll talk, of course, about grunions and parasites. I have another question for you. I want you to maybe think about a person in your life that reminds you of a parasite. Be a family member, a coworker, a friend. All right, so here's the thing. Parasites usually have a bad reputation, right? And that's fair. So if you think about in biology, when you think about parasitism, it's a type of symbiotic relationship. And that's the one where you have the symbiont or the parasite, the small species, harming the host, right, by definition. So usually a lot of times those parasites are associated with disease, right? So they can be causing disease. And you don't think much about parasites unless they're causing some kind of trouble, right? That's when you really hear about them. Knowing all that, this is what my wife and I decide to do in our free time. So a few years ago, we were in Japan for a conference, and in our free time, we decided to visit the Meguro Parasitological Museum in Tokyo. Some people like to go to see art in museums. We like to go see parasites, and they had some really cool ones. I want to share one of them with you guys. That's one of the ones that cause problems. This is a tapeworm, right? It's a big one. If you look at the name of this tapeworm, is Dibothriocephalus nihonkaensis. Are you kidding me? The name is almost longer than the parasite, right? You can see it's a big one, but we can break it down. So the name here means two-grooved-headed worm from the Sea of Japan. Very descriptive, right? So you can see this awesome tapeworm in here. It's really long, almost 30 feet long. So to put that in perspective, that's about the length of 2 Honda Civics. And this parasite is only 3 months old. So it grows really fast, right? This parasite was collected from a human. Yeah, and I know I look very proud and happy in this picture, and I'm pointing to it. I want to make this very clear. It's not from me, but if you read the sign there, it tells the story. Somebody ate raw fish. They ate raw salmon. Everything was fine for 3 months, no symptoms, until one day that person was going to the bathroom and felt something wiggly there. So they immediately contacted the director of the museum. He's like, I can help you. I'll give you some anti-parasitic drugs. And they got it out, and now it's on display, right? So you can go and see it. By the way, the little rope that you find right next to it, right here, you can take it off the wall and just extend it to see how long that parasite is. So really cool. Julie thought it was really cool also. She was super excited about this. All right, so those are the troublemakers. And like I said, it's fair that we think of parasites as troublemakers. But I want to tell you, there's more to parasites than that. So I want to give you a slightly different perspective in here. One thing that most people might not know, parasites are extremely common. You find them everywhere. So of course, the parasite came from a human, but you can find parasites in all ecosystems. Terrestrial ecosystems, aquatic ecosystems, they're all over the place. In fact, if you think about it, you find more species of parasites than species of free-living organisms. Virtually every free-living organism will harbor some kind of parasite. So they're all over the place. While in the same museum, we're walking around, and I found this really cool display of Dr. Yamaguti's notebooks. He's one of the big names in parasitology, and he has these really beautiful drawings of parasites. And right next to it was some other notebooks showing the hosts, the fishes where those parasites are found. So I want you to keep in mind, not all these parasites are necessarily causing disease. They're just there living their lives in the environment, and you can find them all over the place. There's another really cool study. I want to show you just one image from it, a figure, to show you that parasites are part of natural food webs. So here's a food web that is a little bit complicated, so I'll walk you through. So at the very bottom, you have on level 0 there, those are going to be the primary producers, things like algae or plants. And this is a food web from a salt marsh that was studied by Lafferty and others. Then you go to level 1, you have the herbivores, right? The organisms that are eating the algae. And then the next level 2, now they're eating the herbivores and so forth. And all the lines that you see are connections that you have between or among the organisms in this food web. But you only see here the free-living organisms. So one really cool thing that they did, they went out and collected the parasites, and they added the parasites to this food web. So you might be thinking, yeah, parasites, tiny little guys, right? They might show up in here. So I'll show you what they found. So this is a food web now with the parasites included. As you can see, they are important. They are a significant part of this food web, and we should pay attention to them. So now you know parasites are all over the place, and I want to tell you a little bit about parasites in fish ecology. So parasites, they can tell us a lot of information about their hosts. Sometimes they can tell us what the host is eating, can tell us where the host lives, and maybe where the host is hanging out. So there's a lot of information that we can get from parasites. Very nice review paper came out a few years ago, and they're describing why ignoring parasites in fish ecology is a mistake. And this is common. Parasites are difficult to study sometimes. It's very hard to identify them. They're kind of small. So a lot of times fish ecologists, they collect the fishes. Once they find a parasite, they tend to ignore them. Of course, parasitologists know about them. I was lucky to have some great parasitologists help me identify some of my parasites, and I'll show you that. But one point that I want to make is like if you study the parasites, you already know that it's not all the time all about disease and harm. So the parasites sometimes can give us some very important clues about aspects of the host related to both the ecology and the evolution of the host. Speaking of fish ecology brings to one of my favorite fish. Who likes grunion here? Woo! All right, so probably a lot of you guys know, but I want to tell you a little bit about grunion. So the grunion belong to a family called the Tharinopsidae. So those are the New World silversides. I'll share a video. Crossing my fingers, we're going to have a good grunion run tonight. This one was taken a few years ago, thanks to Zach from the aquarium. So here you have the grunion coming out of the water to reproduce. Very soon, you're going to see some of the fish are sticking up. Those are the females, right? They bury in tail first, and they deposit all the eggs. And then the males come and fertilize them. And it's an amazing thing that not every fish species do, right? So it's really amazing that you can come here and see it. One of the things I'll tell you, again, I'm crossing my fingers, maybe you're going to have a good grunion run. But I'll tell you one thing. If we happen not to see a good grunion run tonight, come back. Because they're out there and you're gonna see them. I promise you, like if you keep coming, you will see them. I want to tell a little bit more about the grunion in here. Where do you find them? Like I said, we're very lucky. We live in a place that you can see this amazing fish coming out of the water. The blue line that you see there is highlighting the geographic range of the grunion. It comes from a point down in Baja called Punta Briojos all the way to Point Conception. Sometimes, some years, they can extend a little bit more. So you can see some grunion runs kind of a little further south from Punta Briojos, sometimes further north than San Francisco Bay. I would say the core distribution of the grunion, especially for here where we are, it's right in Southern California. That's where we are. A lot of coastal development, but this is an area that you can always see grunion runs. And again, keeping in mind that we share the habitat with these amazing organisms in here. So I'll tell you a little bit about what you know about the grunion. And I want to highlight a couple of people that I think are very special in studying the grunion. So what do you know about the grunion? Of course, you guys are here tonight, of course, to see parasites, but also to try to see a grunion run. We all know about this amazing reproductive behavior. I think in Southern California, this is a very, very iconic fish, right? If you haven't noticed, there's a fish here in the aquarium logo, and that's a grunion surfing waves, right? So we have in the own Cabrillo Marine Aquarium logo, we have a grunion in here. Now I mentioned in the previous slide that the area can be impacted, right? Because we have a lot of people growing in here, so the population could be declining. At least we have some indication of seeing sometimes a decline on the runs over the years. Now, when I think about grunion, the first person that comes to me is Dr. Boyd Walker, who was a professor at U.S.A. He studied grunion for his dissertation that was done at Scripps in the 1950s, and he gave us a better understanding of the timing of the grunion run. So we're all here tonight hoping for a grunion run. You know, they're related to the tides, and Boyd Walker did a lot to help us better understand that. In fact, the way we measure the grunion runs, there's a metric for evaluating that, and that's called the Walker scale. So here I can show you very briefly. You guys are going to hear more about this tonight. The scale goes from W0 to W5. It's related to the numbers of grunion that appear in that run, the duration of the run, and here you have a description of that, right? If you want to see more details about that, a few years ago, there's a really cool paper detailing everything about the Walker scale. And I want to point out a couple of things. The authors of this paper, they're all here tonight, so you can ask them questions. Yes. We have We have Mike and Suzanne, former directors of the aquarium here. And of course, we have Dr. Karen Martin. Now, when it comes to grunion, I teach marine biology at Saddleback. Nobody knows more about grunion than Dr. Martin. So Dr. Martin is happily retired from Pepperdine University. I mentioned that Boyd Walker helped us better understand the timing of the grunion, right? Dr. Martin helped us understand everything about grunion. A lot of research was done. She published so many articles, a lot of books. This is one of my favorites for you marine biology students. If you're a student of marine biology in Southern California and you don't have this book, you call yourself a marine biologist, you got to go to the gift shop. I think they have it here and get this book. And there's also a great documentary that I always show my students called Surf Sand and Silver Sides. Really cool overview of the grunion and fantastic soundtrack if you haven't seen it. So based on the research that we have with Boyd Walker and Dr. Martin and many others, we know quite a bit about the grunion. We have that window that the grunion comes out of the water and we see them. And we learn actually quite a bit about that. I want to point out, though, that we don't know everything about grunion. And there's a question that we still are trying to answer. And that's the question, who knows where the grunion go, right? I'm going to use a quote from that documentary. It's about the 10-minute mark. And the quote is, little is known about the grunion when they're not on the beach. So hopefully tonight, a little bit later, we might go out on the beach and see something like this. The fish will come out. We have the timing figured it out. We'll see them doing their spawning behavior right there on the beach. And hopefully we'll have a great time. But here's one thing, next morning, maybe you come here at the early hours in the morning and you're gonna see something like this. Now the beach is deserted, like no fish to be seen, and where do they go? So, and one question that came to my mind is like, how can we follow the grunion now? It's kind of tricky, right? So one of the first things that I thought about is like, how do we usually do that with other species, right? How can we track animals that are kind of difficult to follow, or we cannot be there with them all the time? So usually scientists can use tags, right, to track animals to see where they go. A lot of times you hear of physical tags like GPS trackers. You can also use acoustic tags. So if you're trying to find an animal, again, it's flying around like this bird and you cannot find it all the time, or something hard to follow like this hyena, they can have trackers and you can get information about that. Not further from here, if you go down to the Shark Lab at Cal State Long Beach, they do acoustic telemetry to kind of track great white sharks, right? It's part of their beach safety program. So those are all physical tags, right? They're man-made and you need to kind of like put them in the animals and then track them. But a lot of these organisms also have what we call biological tags. So we can use something that the host already has. So some of those are otoliths. Those are the ear stones that fishes can have. They contain some information sometimes about how that fish is growing. One thing that has been attempted also, you can collect some fish. Instead of putting a tag on it, you can cut a little piece of the fin. So that's the fin clip, and then you can see if you can recover some of those. It can be tricky, it's hard to tell. Here's another thing you can do. You can use the host's parasites to track them. And I'm going to show you a little bit how parasites can help us answer some of these questions. So parasites as biological tags. I'm going to show you one of another favorite local fish. Can anybody tell me what this fish is? This is a giant sea bass. And if you look at the head, you see all these little dots, right? Maybe if you saw this picture from far, it's like, oh, interesting, there's all these little dots. They're all parasites. They're all copepods. Those are copepods that Julie studies. And I'll tell you, there's even more. Those parasites have parasites. That's something called hyperparasitism that we're going to save for another talk. But parasites, you can think of them sometimes almost like passport stamps. So the host, or in this case, this fish, can pick up those parasites depending on where they go. Sometimes they might also acquire parasites through diet. In this life cycle in here, the bird is eating a fish and acquiring whatever parasites are inside of the fish, right? And those parasites stay with the host for some time, so we can actually use them as biological tags. Now, this is not something novel. In this book here of stock identification methods for fisheries, there's actually an entire chapter dedicated to parasites as biological tags. So it's something that has been done before. So having the question about the grunion, not knowing where they go, and knowing that it can use the parasites, I wanted to investigate what do we know about grunion parasites. Turns out that we know quite a bit about grunion parasites already, and this is thanks to Dr. Andrew Olson Jr. He was a professor at San Diego State University. I mentioned that Dr. Martin knows everything about parasites, not about parasites, but about grunion, and here she is hanging out with a grunion parasitologist, right? Anybody notice the hat that Dr. Martin is wearing? Cabrillo Marine Aquarium. Woo. All right, Dr. Olson, he studied and reported and described more than 20 species of parasites. So he laid out the foundation for all the parasites the grunion has. So the pioneer work included some aspects of the taxonomy and also some data on the localities where those parasites were found. And this was done mostly in the 1950s through the 1970s. I want to explain one thing about locality. It's like, oh, that's kind of a weird word. Why you're not using location? When you're studying parasites, location is the place in the host where you find the parasites. So if you look at a fish and where the parasite is placed in the fish, that's the location. The places, the geographic places where you find them are localities. So we have a little bit of information about that. Now, it turns out that every good scientific study starts with something that has been done before. So I remember when I started studying grunion parasites, I dig in and I study everything that Dr. Olson did. Turns out that Dr. Martin is also involved with newer parasitologists studying grunion parasites. And this is Dr. Martin, me, and her husband, Doug Martin, in Mexico, right in front of Karen's taco stand. All right, so knowing that, I had the idea for my dissertation study. We had questions. We knew some of those parasites, the taxonomy. We also knew a little bit about their localities, but there are still a few things that we didn't know. So I had 3 questions that I was trying to answer. First question, do parasite communities vary across localities? Do they change geographically, specifically about the California grunion? Another question, do they reveal anything about the grunion movement? And finally, I wanted to know if we compare species related to the grunion, how would their parasite communities change? So this led to my dissertation. So this was a study that I did when I was at UCLA. So I was looking at the Metazoan parasites of the California grunion and also some related species. So with those questions, I set up my project with 3 goals directly related to those questions. So the first goal, to compare the parasite communities across localities. So this is within only one species, just looking at California grunion. Then I wanted to see if by studying those parasites, I could infer anything about the grunion movement. And then the third goal was to compare the parasite communities across the related species. So I went back and started looking at all the parasites that Dr. Olson found. I love this figure in here. It's one of the figures that he used as teaching materials, and you have a grunion and all the other parasites that he had found before. And a lot of us, even though we love grunion sometimes, most of us didn't even know the grunion had any parasites, let alone this many, right? So I'll tell you a little bit about these parasites in here. They're a little bit different. So first I want to talk about this type of parasite, ectoparasites. They usually, we can think of ectoparasites as the parasites that you find outside the host, but by definition, those are parasites that are in direct contact with the outside environment. So I'll show you a few of them here. We have things like copepods, isopods, branchiurs. Those are all types of marine crustaceans. We also have a monogenian here. This is a type of a flatworm that you find inside the gills. So even though it's kind of tucked in inside of the fish, it's still in direct contact with water. So we consider that an ectoparasite. Then you have the other parasites that live inside the fish that you would not even see it if this was a real grunion. Those are called endoparasites, right? The internal parasites. A lot of times those parasites are acquired by diet, right? The fish eats something, now it's inside. So those included different types of flatworms, some digynians, some roundworms, some nematodes, and also some cestodes. So quite a bit of parasites right in the grunion. And this was a great starting point. I remember going back and trying to learn everything about them, but now I wanted to see what these parasites really look like, in person. So I went and collected a bunch of grunion, and I brought them right back here to Cabrillo Marine Aquarium. So I spent some time getting familiar with those parasites, the ectoparasites. I was looking outside. I was looking at the body, the fins, the gills, the oral cavity. Can you guys spot some parasites there? Yes, a few of you. There's a guy hiding here right behind the fin. There's another guy there. We can take a closer look in here, and this is what they look like. So this guy here is Argillus, is one of those branchiurians. If you were scared of parasites, this is the cutest thing you ever seen, right? So really nice. We have copepods here hanging out outside, and this is one of my favorite videos of the monogene right on the gill. You can see a little bit of the gills in here, and he's kind of happy there doing some crunches right there. Then I had to look for some of the internal parasites. So those are endoparasites. So I'll check all the viscera of the fish, right? And I would find things like nematodes and some of those flatworms, some of them right outside the GI tract and some of them inside of the grunion intestines. One side noting, usually for me to go over one single grunion, it takes a little bit more than an hour. So it's a very kind of careful type of inspection, so you can take your time and find everything about these parasites here. So cool, I got familiar with these parasites, and now it's time to collect. Was out to go out in the field, collect grunion, and try to see if those parasites could help me with the questions that I had. All right, some of the field work that I did in here. I ended up and collected 30 samples of 30 specimens at 5 different localities. Now you know the localities are the geographical places where you collect them. I'll highlight them here for you. Pacific Beach down in San Diego, was Seal Beach just down the road, and Cabrillo, like close overlap there as a suggestion of one of my dissertation committee members. Like, it would be nice to do the comparison with some close located spots. Malibu Beach and Goleta, right? So I went out there and collected about 900 fish. And to try to preserve the ectoparasites, the ones that are outside, the first thing I did, collecting the fish, I would immediately bag them and freeze them, but I would bag them to prevent the loss of those ectoparasites, right? So they can very easily fall off. So now I end up with a nice collection of my fishes in here. Now it's time to get data, right? So now I had to dissect all those parasites and collect the data. I'm going to use a couple of terms here. You don't need to worry about it. I'll walk you through. So first, I had some quantitative descriptors of those parasites populations that I found in grunion, and I'll explain a few of them for you. There are things like prevalence, mean intensity, and mean abundance. I'll walk you through the definitions. They're not that difficult. But a lot of times people might ask you which species is more infected, or which fish, and that's a question that is a little bit hard to answer. Is like, which of these parameters are you talking about? So that's why you have this quantitative descriptors. Simple example, we're going to start with 5 fishes, and you can see the red dots, they're parasites in here. So the prevalence is the percentage of the hosts that are going to be infected with any number of parasites. So we have a sample of 5. 3 of those fishes are infected. So what would be the prevalence? You guys are acing it. So 60%. Mean intensity, you only look at the infected fishes. So we have 3 fishes with parasites in here. You count all the parasites, you have 10 parasites in here. So your mean intensity is going to be about 3.3. Mean abundance, similar, but now you consider all the fishes. You see you have the same 10 fishes, same 5 fishes, the same 10 parasites, but now your mean abundance is 2. Keep in mind that right here I'm using the term population because I'm only using one species of parasite, right? Maybe for you guys that maybe remember ecology, like if you're dealing with only one species as a population, If you're dealing with more than one species, now you have a community, right? I also constructed a configuration of parasite communities. I'm going to show another example with 5 fishes, but now those 5 fishes have different parasites, right? So now I'm dealing with a community. If you remember, I collected 30 samples of 30 fish, so I ended up with 900 grunion, right, and data on parasites. So that created a very complex data set. So with those 900 individual fish, sometimes if you're just looking at a spreadsheet of all the data, it's kind of hard to see. So I'll show you a couple of things that I did. First was to have a visual plot that was a little bit easier to make sense of those parasites. I'll walk you through it. You don't need to worry about this, but I did some visual analysis with a type of plot called an NMDS. So this is a non-metric multi-dimensional scaling. And I'll walk you through when I show you this graph. And then, of course, not to have just the visuals, I did some statistics to back it up. All right, so this is a summary of what I found at first. So you're seeing the rank abundance of parasites you find in here. So each one of those bars is showing one parasite species and the total number of parasites that I found. So we're looking at only one species of host, right, the California grunion. I looked at 900 total fishes, and I collected close to 3,000 parasites from them, right? So you can see the distribution of those parasites. The one that I found the most was a copepod, Bomolochus. Then we have the monogene that is found in the gills, nematodes. So I found several different species of parasite. I'm going to show you one thing. A lot of times people ask me, like, how heavily infected was the grunion? I'm going to do something a little bit different here. Usually you like to give those numbers by individual species. If you're doing a study, you look separate at all the different species of parasite. Here I'm going to give you an overall prevalence. I'm combining all the parasites, and I'll tell you that if you take all these parasites into the picture, about 85% of all the grunion that I inspected had at least one type of parasite, right? That's a question that sometimes people have. And the other question is, like, how many parasites did you find per grunion? That brings us to that mean intensity that I told you, and overall about 3.8. So close to every infected grunion that I saw had about 4 parasites in it, right? So that gives you an overall idea. Those are not all the parasites. You've seen the ones that are more common, but you see very quickly these numbers start going down, and you have a long tail of parasites that you only see once. So I have a bunch of ones in here. So I have many more parasites that I found in there. I found a little bit over 20 different species. And to me, that's kind of amazing because, like I said, like a lot of people think like the grunion even have parasites, right? And yes, they have a lot, and they're still out there. So there's all those parasites. All right, I want to show you how these parasites are distributed in a map so you can have an idea if they can tell us where the grunion go. I'm only looking here at 4 of the most common ectoparasites. Remember those that are found outside? And I have here in this box here a little key for the different species. Maybe you start with a cute one, Argulis, the blue one. If you go to the Goleta Beach, you see this is a prevalence. So each bubble in here, it's showing different species of parasite and how big the bubble is. It's how common or how prevalent the parasite is in that locality. So Argulis, about 20% prevalence, so 1 in 5, right? I would find here. You go down to San Diego, the number goes down drastically, so less than 2%. If you look at Caligas, this is a copepod. The prevalence here is very low, 2.8, but if you go down to San Diego, it goes up to 23. So you can see the monogene here, very high prevalence, and then it decreases. So you can see some difference on how common those parasites are in those 2 localities. Then I had my middle ground localities in here. And all those numbers were a little bit more in the middle. They're all intermediate. So it seems like San Diego is different from Goleta. And all those here are a little bit more mixed. But again, I'm only looking at those 4 species of ectoparasites here. Then I did something similar for the endoparasites. Do they tell a different story? Let's take a closer look. So same here. We have a key with the endoparasites. One thing that popped out right away, this is one of the most common ones. It's a digenion you find in the guts of the California grunion. They're found in San Diego. You go up here, they disappear. So they're not there. Some of the other parasites, you can see very low prevalences in San Diego. You go up, the prevalence goes up. So similar to what I saw with the ectoparasites, I saw here now with the endoparasites. And again, in the middle, a little more mixed. The numbers are a little bit more intermediate. But again, each one of those graphs I showed you, it was only 4 parasites at a time. What if we combine all of them? So what if we have a party now, right? We have all those 20-something parasites, and we'll try to make a graph out of that. So that's what I'm going to show you next. That's the MNDS plot that I told you about before. I want to walk you through here. This plot has 30 dots, right, with a label. Each one of those dots represents a sample of 30 fish, right? So the closer the dots are to each other, represents a more similar parasite community. Does that make sense? So in the further away you are, those parasite communities are really different. You're seeing different colors here also. Those are the localities where I collected them, so I can color code this. And the arrows are the parasites that are kind of driving the type of pattern. But there's one really cool thing about this. I remember I plotted this at first, and I want to point this out to you guys. The pattern that we're seeing here, it comes entirely from the parasites. There's absolutely no geographic data in it. It's only parasites, how they group, and how that the different parasite communities are different. So I'll put this on top of a map so you can see it a little bit better, and then the pattern becomes a little bit clearer. I'm going to color code my localities in here so you can see. Goleta, the purple one, is right here on the top. If you look at San Diego and Pacific Beach is right here on the bottom. You see the parasite communities between Goleta and San Diego, they're pretty different. There's no overlap in there. Imagine if I don't have the ones in the middle, I'd say, wow, yeah, the grunion parasites in Goleta and San Diego, they're pretty different. In the middle, you see more of an overlap, right? And the sites are closer and the parasites are more similar. So in a way, when I looked at this, I said, this is really cool. This is telling me a couple of things. One, the grunion is not freely moving within the Southern California Bight, because if they were, you would see a mix of all these colored dots all over the place, and we're not seeing that. We're seeing basically 3 main groups in here. So one down south, one north, and one in the middle, right? It has something a little bit more common. So that is telling us that the grunion is probably staying close to the places where we see them spawning. I also found some species that were new records for the grunion. They're actually not located super close to the beach. They're a little bit more pelagic, a little deeper. So that tells us a little bit also that the grunion might be moving all the way out, right? But again, not a whole lot of mixing, and you have more separate parasite communities. So this is for only one species. I was only looking at the California grunion, but then I wanted to go and investigate the relatives of the grunion, right? To see how does that compare. There's at least 2 other species in the same family that we find right here. If you go outside the beach here, you can find them. So I'm going to go back and look at the parasites of the New World silversides, that same family that the grunion is from. So I'm going to be looking at 4 species. 3 are found right here in Southern California. So California grunion here on the bottom, and you also have top smelt and jack smelt. They're all found. Again, you can go out of the beach here, you can probably find them. There's one more species that I wanted to add and compare. Didn't know if you guys knew this, but the grunion has a sister species. And this is the Gulf grunion. So it's found in the Gulf of California in Mexico. So want to focus on those 4 species for now. So here we have the 2 grunion, the 2 jack smelt. Again, I told you the 3 that are going to be on the left side for you are found in Southern California, and the Gulf grunion is found in Mexico. Can you guys kind of see where they are placed? I think a map would help, right? So we have a map here. All right. So for the ones that are found on the Pacific side, all those 4 fishes are part of the New World silversides, but there's a little bit more to them. When you look at their evolutionary history, they're a little bit different. If we look at those 3 species in here, they diverged almost 18 million years ago. So they live in the same place, but they are a little bit different. They're not super closely related. If you look at the two grunions, in terms of evolution, they're the closest. They're the ones that share the most recent common ancestor. They both are in the same genus, and they look almost exactly the same. So they have been isolated for about 1 million years by the Baja California Peninsula, right? So so those two species they never come close. This here is the north part of the Gulf of California, also known as the Sierra Cortez. If you keep going down the Baja Peninsula here, you're going to end up in Cabo. The Gulf grunion doesn't go all the way south there, so they stuck a little bit closer to the north part. So they're never in contact with the California grunion, even though they are the closest relative, right? So to make this comparison now, I need to go and collect more fish. Right, so those yellow dots here are my collection sites. OCB is Outer Cabrillo Beach, right outside. This is Belmont Shores. We have San Diego Bay. But for the Gulf grunion, I had to travel, right? So I had to go and take several trips down to Mexico. I want to take a little side deflection here and tell you about the collection of grunion in the Gulf. This was not easy. It required a lot of planning. So I took multiple trips there. I had some fantastic help. So a lot of people came and tried to help me collect the grunion. We have Bain here from the aquarium. He's here in the back. Dr. Ralph Epi came to a couple of those trips. My friend Dr. Kimo Morris and Dr. Jeff Watanabe, they came also. But besides all those trips and the help, we also have some really great stories. Each one of those pictures have a story behind them. I remember maybe in this one, there was a dead whale on the beach or something crazy like that. It was really cool. Kim and I had a horrible misjudgment in eating a seafood volcano in Baja, and we both got super sick. That picture there with Dr. Ralph Epi has a good story also that maybe I'll save for some other time. We got our car stuck in the sand, but there's another story. There's a story that I wanted to tell you. I was getting frustrated because I took so many trips. It takes at least like 6-7 hours to get to the places where I was trying to collect. And I couldn't find any grunion. And I was asking people there, I was like, in Spanish, they call pejerrey, and I speak a little Spanish. So I was like, I'm looking for pejerrey. And it's like, oh, no, they come here, but only during Easter time. I was like, no, I know. I have all my scientific journals, all the timings, and it's like, they should be here. And I couldn't find anybody who knew about the Gulf grunion. And then one time I met a security guard that worked on the night shift, and he was the only person that I met there that knew about grunion, right? And he's like, oh, no, I see them. I'm working here and I see the grunion coming. And I was like, wow, that's great. You're the first person that I mean here that knows about the grunion. And I was like, when do they come? He's like, oh, that I cannot tell you. I don't know, right? It's like, come on, man. I was like, okay. I was like, does anybody know when the grunion will come in here, right? And he looked at me, he's like, well, you want to know for sure? I was like, yeah, I want to know for sure. I'm doing this research. I need to collect them. And he's like, well, if you want to know for sure, Satanas knows. And I was like, no. I remember I speak a little Spanish. I was like, Satanas. I look at Bain. He's like, is that Satan? And Bain said, I think so. I was like, oh, that's kind of creepy, right? And Bain was looking at me. He's like, how bad do you want this fish? You know? And I was like, I don't know. And Bain and I were talking, and then the guy said, look, Satanas is here. I was like, yeah, I don't know if I need this. And then I saw it. I was so relieved to see Satanas the cat. And he's like, every time the grunion comes out, Satanas. This is a picture of the first Gulf grunion I ever seen. Like, we're right there. I was relieved. I was like, oh my God, this is awesome. It took another trip. It took a little longer. We came down one more time, and then I finally saw my first good grunion run in the Gulf. Has anybody here seen a grunion run in the Gulf? Maybe a couple of you guys. I'll show you what I saw. So first thing that you guys might be noticing here is that it's daytime. So this is different because the timing of the tides there is a little bit different. And depending on the time of the year, you can go there and see the grunion running during the day. Now, the thing you might notice, this is kind of protected, right? So not a lot of nice surfing waves like we have in here. So tiny little wind waves. The grunion stays right there on the edge. It's very hard to collect them. When you approach them, they all disappear. But we're able to collect them, and then I could finish my research comparing these parasites to the local species in here. So I'll show you what I did with that data and what I found here. So we'll go back here to the parasites of New World silversides. Question for you guys. A few slides ago, I showed you my one slide with the California grunion parasites. I had the number of hosts. I had the number of parasites, the prevalence, and the mean intensity. Do you guys remember those numbers? You remember the slide, but you probably don't remember the numbers, right? I know that. So here are the numbers. So you can refresh your memory. So I look at 900 California grunion from the previous study. Close to 3,000 parasites in here. Overall prevalence was 85, and now the mean intensity was at 3.8. So now I'm comparing all 4 species. So the total number of hosts that I looked was about 114, and you can see the distribution, how I had about 30 hosts of each species. So maybe you think, yeah, you got lazy, huh? After all those 900 hosts. I'll show you, not really. I'm going to show you the total numbers. So if I got 900 hosts first in 2,000 parasites, this time here with about 100 hosts, I got almost twice as many. So there's a lot more parasites when I combine all the species. And I'll show you how those parasites were distributed in here. The overall prevalence, I never seen a top smelt, a jack smelt, or a Gulf grunion that didn't have a parasite. So all of them have at least one species of parasite in here. And remember the California grunion mean intensity of 3.8? We can see how this guy's comparing here. So yeah, a lot more, right? So top smelt on average, every infected top smelt, which is all of them, had on average 76 parasites in them. For jack smelt, over 100. The Gulf grunion had a few, and the California grunion with that smaller sample was close to the other study that I did. So you can see that what is really driving this difference in parasites are the parasites that I'm finding in these 2 species in here. I'm going to give you a snapshot in here. When you look at the guts of top smelt and jack smelt, this is what you find. They're loaded with parasites. There's a lot of like larval tapeworms, lots of nematodes. Yes, that's right. And you see them all over the place in here. I showed you a slide with the rank abundance of the parasites of the California grunion. I'm going to show you one here with all 4 species combined. The difference here is that because I have 4 species, I color-coded each of the bars, right? So here you can see this is the parasite that was the most common. And you can see all this top 3 most common parasites came mostly from those 2 species in here. So, and then the Gulf grunion had a few, the California grunion, but most in terms of numbers, man, top smelt and jack smelt. I don't know if you guys ever saw a grunion and a top smelt side by side. They can be hard to tell apart. If you study them, like I can tell them right away, but even if I give to somebody who studies fishes, and they look like the same fish, but they're very different in their parasite compositions. So this table here is showing the most common endoparasites and the most common ectoparasites here. The ectoparasites are cool, but the numbers are low. The endoparasites are really driving this here. So you have nematodes, a larval tapeworm, and you have another larval parasite in here. You can see this is really driving how those parasite communities are. So I did something similar that I did with the grunion. I want to get a visual plot of it. Bless you. And here we have the 4 species on that NMDS plot, but this time was a little bit different. Instead of using each point as a sample, now each point is a host, is a fish. So here, each individual fish is a point, and all the parasites that they have inside with their communities. So one of the first things you see in here, you look on your right side and you see the 2 grunion species. They're not overlapping, but they're all on the same side of the graph. And then when you look on the jack smelt and top smelt, there's some heavy overlapping in here. But one thing that popped out a lot to me is that The California grunion lives right where the jack smelt and the toss smelt is, but the parasites are very different. In fact, when I look at the type of parasites, they're a little bit more similar to the Gulf grunion. And I was thinking, what can cause that? If you look at the diet of those species, they're very different. Jack smelt and top smelt are generalists. They'll eat everything out there. They're omnivores, and they're probably acquiring a lot of those endoparasites by diet. The other two species, they're specialists. The California grunion and Gulf grunion, they feed almost solely on mice and shrimp. So they're getting a lot of protein, probably will help them come to the beach and reproduce, but they're not acquiring nearly as many of those parasites. So even if you look at them side by side, they look the same, but the parasite communities can be pretty different. All right, a couple of things to summarize what I just told you. One, that I want you to remember, parasites, by definition, they can cause harm, but that's not the entire part of the story, right? They can be much more than just harmful. So when I looked only within species, the California grunion, I saw a little bit on how those parasite communities were different along the coast, right? It's telling that the grunion is not mixing that much. So the parasite communities in this case, it's telling us a little bit about where the grunion go along the coast. When I decided to look to the other species, I found that even when a fish looks almost exactly the same and lives in the same area, the parasite communities may still be different, right? So that's telling me a little bit more about the evolutionary history of the host and how, as they develop over time, they feed on different things, and their parasite communities are different. One of the things I want to tell you, after all this work, I want to remind everybody that you never can do this much work alone, right? So I have a few things to acknowledge here. I really want to thank my dissertation committee, my advisor, Don Booth, Ralph Eppy, and Dr. Karen Martin, like they're part of the committee, as well as Dave Jacobs and Michael Shin, Julie Passarelli for her support, and also identifying parasites. Julie is a copepod specialist, and Danny Tang, Ralph, is Ralph here tonight? Thought he was going to come. Ralph is a parasitologist of, man, there's no comparison. He knows a lot about all different types of parasites, and he helped a lot. So we started with a question and want to bring this question back here to you guys. And now with parasites in it. So do the parasites know where the grunion go? So if you really think about, not exactly the parasite don't know, right, consciously, but they can tell us stories. If you think about each parasite that you find in a host is a record, and that record can tell us where a fish has been. It can tell us what a fish has eaten. It can tell us what the fish do in terms of interactions with the environment and also with other species. So they're not just harmful, they're actually little storytellers. And the next time you go out on the beach and you see a grunion run, I want you to think about that each one of those fishes have a story behind them, and those parasites are helping us to discover those stories and actually to make new discoveries. Thank you.
Thank you, Bruno. That was awesome talk. We have some time for questions. Let me just remind everybody, I guess I forgot to mention this. If you notice, we do have these discs up on the ceiling, and now the lights are green. That means hot mic. So everything that you guys say, the folks at home can hear. So they'll be able to hear your questions, but if you're chatting with your neighbor like, oh my God, those parasites are nasty, everyone at home is going to hear that. So just FYI. So I'll let Bruno answer a few questions, and then I'll see if there's some questions from home and see if they're typing them, and I will read those out. But why don't you take some from the audience?
Hi, thank you. First of all, it's great. Is there anything interesting you can learn from all those ones that you had in that 900, when you're looking at those 900, just the California grunion? Is there anything that you found like more of the Goleta Beach or Pacific, or the ones in the middle were having kind of those specialty one-off parasites, or is there any information you could glean from that? Or was that just kind of more, was that more random that?
Yeah, that's a great question, right? So those ones are rare parasites, right? They're not the ones that are very common, but you can learn from them also. There's one of the parasites I might have mentioned briefly. It's a parasite that we find way further out in the pelagic environment, that it's not where typically you would think, oh, a grunion is close in a kelp forest. No, it's way out there. And of course, if you have more numbers, you can make better inferences, but they give us clues. Again, I don't feel sometimes they get a perfect answer, but everything's a piece of the puzzle.
Yeah,
So,
yeah, okay, cool.
yeah, yes. The rarest parasite. That's a tough one. Like one thing I'll tell you about the distribution of those rarest parasites. I can tell you the most common one, right, which was a copepod that he found. But the rarest parasites, as you see, in a way, every time you look at a big sample of fish, you find the most common parasites, and there's a few of them, but those numbers start dropping really quick, and you end up with a big tail of rare parasites. So there's many of them, and that's typical of the distribution that you find in the fishes. But because there's so many different ones, it's kind of to tell which one is the rarest, right? Right.
Is there any any other like state or country where they have grunion runs and they like, yeah?
That's a great question. So, as I mentioned, we're very lucky to see the grunion runs. The California grunion will only run here in California and down in Baja. So, it's something that we can only see here in California and down in Mexico. The other species is also in Mexico, but not on the Pacific coast. They're inside the Baja Peninsula, so that's in the Sea of Cortez. But those are the only two places that you can see the two species of grunion, and nowhere else in the world. So that's another important reason why we should do the best we can to conserve it, right? Because they're so special. Yes.
Do you know what the evolutionary history is of those four species? Thinking possibly that the grunion are late arrivals and have selectively chosen a different series of foodstuffs to use based on coming later and more specialized from, say, the jack smelt, or I don't know which ones are evolutionary first.
Yeah, no, that's a great question, and perhaps even probably Ben Higgins and Michael Horn, who did the whole study on the diet of those four species, could answer that. But in a way, if you look at organisms in the marine environment, you have all these specific niches, and they're all trying to adapt and have their own space so they can, like you said, maybe compete for limited resources. So they all do a few different ways. I think both in a combination of the paper with the diet and the paper that really explaining when that split kind of happened in terms of the diversion of the four species, probably then you can get a better idea, a clear idea of when that happens. But in a sense, you're right. They're all evolved to kind of use the environment and acquired food differently. So now they end up with different parasites. I think in the part that I'm interested is more looking at how the parasites can be related to the diet, right? In a sense, because yeah, depending on what you're eating, you're going to get different things. Yes.
For the California grunion, is it possible that the parasites might die or leave the host as the grunions move up and down the coast? Like as the water quality, the season changes, or life cycle or something?
Yeah, no, this is it's a great question in here. A lot of times we hear that a healthy ecosystem is an ecosystem that has parasites in it, because as we saw in those food webs, they are part of the environment. One thing that I didn't go into a lot of details, a lot of parasites have very complex life cycles. They have multiple hosts. So if you lose any part of the life cycle, then yes, you could lose the parasites like you mentioned. So if an ecosystem is very healthy and all the hosts are present, then the parasite can complete the life cycle, and you could see that. But yeah, you could see changes happening in the ecosystem where you start losing parasites. And depending on the parasite, that there's a lot of different nuances to that. Like you can get an idea on how it's affecting the species. But yes, yes, you had a question.
Yeah, has there been any studies regarding the parasites on any like of the outlier runs, like further north or more southern?
Great question. As far as I know, no. And actually, I wanted to take a peek on those because they're kind of rare, right? What's happening there? And they're almost like one-offs. Probably Dr. Martin can tell us more about that, when those runs happen, if they're tied to El Niños or things like that. But I would be very happy to take a look at some of those those grunion that appear up north. No, I don't think there's any studies that have been done there. Yes.
You mentioned at the beginning of your presentation that even parasites can have parasites. Did you observe any cases of this with your specimens?
With the grunion, I don't think I have any of those examples. The first one that comes to mind is the one that I showed you, and I know that Julie studies them. So some of those copepods, they have more parasites. What are they?
Yeah. Yeah, so right now we have 3 examples that we have a student that is going to be studying that for their master's. And we found them all on copepods, on giant sea bass, giant kelpfish, and different types of elasmobranchs. And it's a type of monogene that lives on the copepod. And there's only something like eight species of that monogene known in the world. So it's a very rare thing. But they live on the back of the copepod that lives on the fish. Yeah, but we don't think it's feeding on the copepod. We think it's feeding on the fish. But to TBD. How about we ask a few?
One more question.
I like sashimi. How do I avoid tapeworm?
What is that? I couldn't use. Oh, don't talk to Julie, because
What's the purpose of parasites in nature? Do they have a purpose?
oh, great question. I think that the purpose of parasites in nature is the same purpose that we have in nature. We're finding our own space in the planet, and we're just existing, and the parasites do the same. One of those things is sometimes like, well, we're a lot more evolved than parasites. Not really. If we're all alive here today, we're all made it, and we're all equally evolved in a way. So, and if you think about it in terms, I wouldn't say in terms of the purpose of the parasite. I don't want to get too much into the meaning of life of parasites, but if you look at those food webs, for example, you can tell even by looking at those lines, right, showing like even visually, like they're a huge part of those food webs. I'll tell you this: if you remove the parasites, you won't have the same salt marsh or any ecosystems that you have now. So in a way, their purpose is to keep everything in balance the way these ecosystems evolved. So they play a very important ecological role.
Okay, so let's get a couple questions from home.
Okay.
One is from Carl. He's up
Hi.
north. Hi, Carl. Are any of those endoparasites using grunion as intermediate hosts?
Oh yes, and if we go into all the different details, like there's very specific life cycles. A lot of those endoparasites, the grunion is an intermediate host. So you have something else that could be a bigger fish. It could be a bird that is actually eating grunion. So yeah, many of them. There's quite a few examples. One thing that I was going to go back just to the sashimi question really quick. I love sashimi too. Julie's vegan, so she won't touch them. But I'll tell you this: if you eat sashimi or sushi, and I tell this to my students, make sure you get it from a place that is safe. If they usually, if you have raw fish, jokes aside, if you cook the fish, that should kill the parasite, and you're fine. I was talking to some folks earlier. Oh man, I eat grunion and all those parasites. Like, yeah, did you cook it? That's extra protein. That's fine. So everybody's about protein right now. If you like sushi and raw fish, you want to make sure that it was frozen to a temperature that is sushi grade. And like I tell my students, do not buy sushi at gas stations.
Got your fish. Got it.
That's right. You can
Yeah,
cut it.
got it. So you get on the terms like that right away. Okay, Amanda asks, did you notice any temporal changes in parasite prevalence or intensity?
Great question. When I collected California grunion, it was for 3 years. I think collected 2016, 17, and 18. And I didn't see a lot of differences there. I remember there was part of my project that I wanted to look at temperature changes or some other environmental data, but I don't think I had the right kind of temporal window to see those changes. But it's been 10 years since I collected those fishes, so it will be interesting to see if there's any changes now.
Okay, how about one more question?
All right, Leonardo.
Yes, great presentation, really.
Thank you.
But I had a question. You mentioned about parasites not being harmful necessarily. Did you find examples in your sampling of commensalism or non-harmful relationships?
Fantastic question. And a couple, by definition, the parasites harm the host, right? That's the definition. So if you start going into commensalism and mutualism, those are different types of symbiotic relationships. If the symbiote is causing harm, then that by definition, that's a parasite. A couple of things to keep in mind. Causing harm doesn't necessarily mean killing the host. If you think about it, if the parasite kills its host, it's losing its place to live sometimes, right? So a lot of the parasites that I'm seeing in grunion, by definition, they're harmful, but some of those copepods are maybe eating skin cells on the outside. Yeah, it's consuming resources, but I haven't seen in the fishes that I collected anything that's like, oh man, these fishes look very sick or unhealthy. They all look healthy and they have parasites. So in that sense, the level of harm can be different, right? And again, one of the things that I wanted to point out also is that by definition, parasites are harmful. There's lots of like horrific examples of diseases and things that if you think worldwide, like malaria and other things that actually kill a lot of people, and parasites can be really serious. One of the things that I wanted to show you is that parasites can help us answer some questions, so they can be a little bit more than just harmful, but also keep in mind that the level of harm can vary quite a bit.
Okay, thank you. We can all have a round of applause.